What do you think, Patrick? Should we start?
Yeah, this is a pretty good showing. And it is a couple of minutes after 4:30. So I see Ben, my old roommate from the very first Ocean
I
Memory
know we've got
Workshop.
some old comers and newcomers here. This is great.
Yeah.
Hey, hey,
How you doing?
doing all right. How about you?
We're doing great. It's good to see
Good.
everyone.
Wow.
All right, I guess we'll get started. Welcome everyone to the first ever Ocean Memory webinar. This is on evolution, communication, and memory in the microbes of the ocean. And we thought this would be a great way to get people together during this kind of time of uncertainty and not knowing when we're going to be together in person again. So at least we can share and learn from each other and be together in this capacity. So we're going to, I'm going to introduce John. And before I do that, I just wanted to say he's going to speak for about 20 minutes. And then we're going to have a couple of minutes for maybe some immediate reactions to that. Either you could just unmute yourself and speak up, or you can type something in the chat. And Patrick will moderate that quick discussion. And then we'll introduce Rika. And then after Rika's talk, we will have about 15 or so minutes to talk collectively. And then after that, there's going to be, there's already a Slack channel set up. It's in the chat. If you wanted to continue the conversation, we're going to continue it there. So that is the format. And I don't want to waste time, precious time here. So I'm going to get right started. Can you guys hear me? All right, sorry. John Barrows is an oceanographer and astrobiologist at the University of Washington in Seattle. He has particular interest in the microbiology of extreme environments and the significance of early Earth history and submarine hydrothermal vent systems for understanding the origin and evolution of life and the possibility of life on other planets or moons. The ocean's memories of early life, viruses, and the evolutionary process deeply influence his thinking across these areas of interest. At UW, he has trained many graduate students, including Rika, and published extensively on these subjects. He is one of the founding faculty members to establish the nation's first graduate training program in astrobiology. And he has served on more than 40 national and international committees, including as chair of the National Research Council, and so forth. And we, without further ado, let's introduce John.
So I'm John, and can you see my full screen? So I'm gonna see, trying to hide these. Okay, so first of all, I would like to thank the organizers for actually inviting me. They probably didn't expect me to thank them for this, but I am. And also for those of you who are attending during these really stressful times, I really appreciate your doing that. Rika and I came up pretty much with talking about evolution, communication, and memory in the microbes in the ocean. And we will be sort of in a lot of ways complementing each other on what we're going to say. So the topics for discussion primarily are what are the ocean's earliest memories, and how did the origin of life alter these memories? And now I'm going to put a lot of emphasis on really the earliest ocean, and I'll talk a little bit about that and when the ocean was formed, when life was formed, etc. And then I want to take a segue towards the end and talk about a different way of communicating that is between early networks and earliest organisms, even before organisms actually evolved. And Rika will build a lot on some of the things I'm going to say, particularly with regards to the last common ancestor and the earliest cells, how they communicated. And then she'll be talking a little bit about the kind of work that she's doing also, and trying to understand the earliest organisms, their proteins, etc. So my first topic again is, what are the ocean's earliest memories? And I like to go to this slide. If you look at the red circle, we're looking at the formation of crust in the hydrosphere here at somewhere between 4.32 and 4.52 billion years ago, which is during the Hadean. So in this case, what we're dealing with is some of the earliest ocean, and whether or not there's some memories here that we can actually talk about from this very early period when an ocean was formed. So what are the ocean's earliest memories? So first of all, the formation of ocean crust in the hydrosphere is what we're talking about. And during this period, there was extensive volcanic activity, including two types that I will talk briefly about. As a result of the hot magma and water rock interactions took place, circulating many elements vital for life. And I'll talk about that also. A little bit later on, transport of water and rock into the mantle through subduction. And I'll mention how vital that is to maintaining essentially the ocean and its activities. And later on, I'll talk a little bit about the formation of minerals, vital chemical reactions that are involved in the origins. So the properties again of the Hadean Archean period, again, widespread hydrothermal activity. And there are two different kinds, and I'll talk a little bit about these. Magma-hosted type, which is the ones most commonly studied, and I'll talk a little bit more about those. And then the peridotite-hosted system was discovered in 2000, and I'll mention that a little bit. It's a dominant mineral in the upper mantle, and it consists primarily of the mineral olivine, which is a magnesium iron silicate mineral. And the result is that the overall crust, or the basalt, and you can see in the bottom figure of a modern ocean crustal system, the basalt is dominated by olivine-like minerals, particularly during this early period. And then we had early stages of subduction. I have pictures of the two vents that I'm going to talk a little bit about, magma-hosted and peridotite-hosted. And I couldn't resist an artist's rendition of the earliest Archean, but I'm sure some of you artists could probably do a better job on that. So the two types, I'll talk about them separately. First of all, the magma-hosted hydrothermal system. What is really important here is that many elements required for life are produced from water rock interactions, along with some carbon sources, very catalytic minerals, etc. Temperatures can reach greater than 400 degrees Celsius, and at the hot fluids, the pH can be quite acidic. More than 240 vent sites have been discovered to date. So a couple of things that these magma-hosted hydrothermal systems do. They produce carbon and energy sources for organisms, such as CO2 and hydrogen and methane, organic compounds. They're also a major source of elements required or used for life, and I'll talk a little bit about that later. And they form multiple gradients, particularly temperature, pH, salinity, chemical, mineral, that allow a support of a wide range of microbial species, viruses, and animal species. What's interesting about animal species there's mostly higher taxa, uh, which is quite interesting. For example, let me see, what is the, essentially there's very few species to any one taxonomic or genera group, but which is very unusual. So you actually have more than, I think, 700 species and with almost close to 300 genera. Some of the animals I have on the bottom, the famous tube worm that has symbionts running it, which are primarily sulfur oxidizers. The second one is an alvanellid worm. I'm highlighting it because it's probably one of the highest temperature growing multicellular animals on the planet. And it's been, one species actually likes growing at 50 degrees Celsius, and it's adapted to be high temperature based on its enzymes. The third one is a snail, which is a scaled armored snail, which is very, very interesting also in that the top layer consists of 3 different layers and are most resistant to any kind of penetration we know. In fact, the military has picked this one up and is actually looking at it as a way to increase its armor. The fourth one is just one of, as I say, many very interesting things, the Yeti crab, and you can see all the little filaments around it. What this particular animal does is garden. So it grows organisms on its appendages and then eats them, something that we may have to do later on as our population grows. So think about your own body gardening and cropping that off. But anyway, that's just another aspect of, I think, well, anyway, it's weird. So when we get back to what we're talking about, I think one of the most important aspects of this is that approximately 40 elements, key elements, are produced in magma-hosted hydrothermal systems. And the ones highlighted in red are essential for diversity of microbes and higher organisms. And they're involved in as nutrients, energy sources, alternate electron acceptors. Some organisms, microorganisms, breathe some of these in the oxidized states. But look at the number that are involved in metalloenzymes. And these are basically enzymes that have these metal sulfur clusters to them. Many of these are considered to be the most ancient. For example, there's a couple of papers out, including the W and the MO, the tungsten and the molybdenum may be the first group of enzymes ever formed and may be involved very early on in the origin of life. The result of this is a hypothesis that Eva Stucken's paper that also has Rika on it, that mineral catalysis preceded protein catalysts, and furthermore, were the backbone of prebiotic reaction networks that led to modern metabolism. So essentially, we know very little about minerals so far. We have maybe less than 10 that we're really studying extensively, but in terms of ocean memory and what minerals do, we have a long way to go. And so that's ongoing research, and there's some pretty cool people working on it. The second one is the peridotite hosted system, and it's noted primarily for producing, as a result of the chemical reaction, high temperatures, and it also produces high concentrations of hydrogen, methane, other hydrocarbons, and it's very alkaline. And because of its alkaline nature, carbon dioxide precipitates and forms carbonate chimneys like this. It was first discovered in 2000 by accident by a researcher at the University of Washington, and it's called Lost City. And while this is a complicated slide, the take-home message here that I want is that olivine, which is, as I say, the very component of mafic crust, when combined with water, produce high pH levels 9 to 11. It can produce, particularly at the site of reaction, fairly high temperatures, just in the water rock reaction of greater than 250. And it produces serpentine. And serpentine has been hydrated. That is, it has about 30% more water than, let's say, olivine had. And I'll mention this as really pretty important later on. The main take-home message, however, is that hydrogen is produced, reduces CO2 into a variety of compounds, including hydrocarbons and organic acids that are really important. And then recently in 2018, the amino acid tryptophan was found also to be synthesized abiotically. This is an important particular amino acid since it has what we call a heterocyclic component to it, a ring structure that may actually say something about the origin of maybe nucleotide bases. So in summary of this is that it does, serpentinization does really two things. It produces heat and it expands a rock by roughly 1/3 by gaining water. So when plates or oceanic plates undergo subduction, the heat and pressure reverse the serpentinization reaction. In other words, when these hydrated minerals get back down into the magma, they dehydrate and form olivine again, and this comes back up, and we have this cycle. So Lost City carries a huge amount of water down into the mantle, part of the vast and complex cycles that keep the Earth running. Unfortunately, so anyway, I think what is important here, just as a take-home message, is that this again is a process that's over 4 billion years and still going on today. It's a key component to early ocean memory. Come on, it's having a hard time. The second thing I want to talk about here at the end is, in this particular figure, is when life started. We do have some evidence that based on isotopic signatures, microfossils, etc., that life may have actually already been in existence at between 3.4 and 3.7 billion years ago. So these are some of the oldest microfossils. I've sort of highlighted the third one, which is one that is very clearly from hydrothermal environments at approximately 3.77 billion years ago. And again, I think the question that I ask all the time, is there a relationship between the environmental settings of early life and the environmental settings for the origin of life? And some of my thoughts are, when did life appear on Earth? Does the settings where there is evidence for early life say anything about the settings for the origin of life? There's a lot of papers coming out saying the origin of life occurred greater than 4 billion years ago, and that what we're seeing in the fossil record is real life. And so, again, I stress the when, where, what, how questions. Where was the environmental settings where life arose? Was hydrothermal systems dominated, particularly in mafic crust? A lot of original life studies are being done associated with serpentinizing type environments. And what can we infer about the earliest life form? And I think this is something Rika will talk about. And then I think the formidable question that I'm not going to address is, how did life arise from chemistry? And that's basically a whole course of controversy. But one of the things that we've talked about with Rika and other former students here is a possibility that the origin of life involved a global Earth. So the topic 2, to sort of end, get close to the end, was how was information communicated between the earliest networks and the earliest organism. I broadly define communication as the transmission and exchange of information that creates conditions that build greater complexity. So it can be in many different forms. And what I'm looking at here is, this is somewhat analogous to, I think, Darwin's survival of the fittest, reactions and processes that fostered progress that lead to greater complexity. This is something I'd like to develop a little bit more with the Ocean Memory Group. So the key properties of Earth life that merged into living organisms that I'm just thinking about, the elements that are required, various energy sources, catalytic reactions, for example, minerals, etc., and the requirements to be able to oxidize and reduce. And so what we have is a combination of networks, selection, increasing complexity, somehow transmission to the next level, and eventually memory. So the common ancestor to all life as we know it is the result of this. And Rika will talk about that also. I have one more slide really, but Earth life has this common ancestor. I wanted to emphasize what this means. That concept has its origin in what is referred to as the unity of biochemistry. That is, all extant organisms share the same biochemical and molecular characteristics. Same nucleotide bases, same 20 amino acids, same genetic code, lipids such as what's found in membranes, etc. And they use things like phosphate as ATP and thioesters as energy sources. So the unity of biochemistry is really reflected in the evolutionary relationship between all life, referred to as the global as the global phylogenetic tree, which will be discussed in the next talk. So my summary really is oceans existed at 4.4 billion years ago, based on what we call zircon data. I'll answer any questions about that if you're interested. Life originated on Earth sometime between 3.4 and potentially 4.1 billion years ago, probably closer to 3.7 or 3.8 to 4.1. During the early stages of Earth history, particularly the first billion years, the mantle, based on models, was quite hot and may not have had plate tectonics early on. This would all favor volcanism and extensive mafic crust, the kinds of things that we talked about with serpentinization. So the result would be widespread serpentinization, synthesis of organic compounds, with the potential for widespread polymerization given potential gradients. So the global Earth may have been more like a chemical reaction with physical and chemical gradients and lots of hydrogen. So thank you.
Thank you, John.
I'm done.
Thank you. This little interim period is meant to be a place for a quick comment or even like a place marker question for something that you want to ask John now and let him think about, and maybe he can give you a quick answer now or answer at the end, or just to get the juices flowing. Does anyone have anything they want to drop in this little interim time period? I don't see any comments.
I have a quick question. This is Melody.
Hi, Melody.
Hi. So John, during your talk, you mentioned that at the Lost City, there's a large volume of water that goes back into the crust. And I was wondering what the significance of that movement is.
Well, it really is what drives hydrothermal systems, really. And so in order to keep the cycle going, we need, you know, there's different ways that, you know, in modern ocean today, there's other ways, cracking and that water also gets down into the mantle and heat, particularly with magma-hosted hydrothermal systems. But I think the key that I want to made with the Lost City was that what we have is a cycle that we go from olivine to serpentine, and that gets rehydrated again. Get dehydrated again into olivine and gets recycled. What makes this really, I'm really fascinated by this for reasons I don't have time to talk about. For example, one of the moons of Saturn, Enceladus, actually has many characteristics of what we find in Lost City, without, like I say, the complex crustal material in that. And so what we're thinking of is that the hydration, dehydration comes with what we call tidal fluxing, since it orbits at an uneven way. So anyway, I think there's broad ramifications here, and I don't want to get too carried away with this, but
Yeah.
it's a really interesting topic.
Okay, and we have a few comments. We'll bring them up. That's good to see all these comments. We'll bring them up later, right? We're already at the halfway mark. So everyone agree we should move on to Rika now?
Yes, I agree.
Okay. I think, Rebecca, you have the intro duty capability.
Sorry, I was muted and then lost my screen. Thank you so much, John. We're so thrilled that you were willing to speak at our first webinar. And we wanted to follow up with Rika, who's worked with you. Rika is an assistant professor at Carleton College in Northfield, Minnesota, in the biology department. And the overarching goal of her research, is to understand how life co-evolves with its environment over time. She uses next generation sequencing tools to better understand the processes that drive microbial and viral evolution over time. And she collaborates with astronomers, geologists, chemists, and atmospheric scientists to ask big picture questions in astrobiology and oceanography. And I am very happy to welcome Rika.
Thanks. All right, let's see if I can do the share screen here. Yes, awesome. Okay. So I thought I'd start with a shot that I took on the Ocean Memory Cruise, which was happening around this time of year last year. This is taken from Friday Harbor, which is, I think I'm actually sitting like right here right now. I'm on Lopez Island right now, up in the Salish Sea. So I'm going to be talking a little bit about jumping off from what John was talking about. So he was talking about the very earliest origin of life, and I'm going to go into how we learn about the last universal common ancestor to all of life on Earth, and then how we infer something about how life evolved since then using ocean memory, basically. Okay, so I'm going to be talking about Luca. And so Luca is the last universal common ancestor to all life on Earth, like I said. This is a nice image that came out of New Scientist a couple of years ago, showing that this is what I'm talking about, Luca here. And then from Luca came the other branches of the tree of life as we know it. So that includes the archaea and the bacteria, which are single-celled organisms that live in and on and all around us. And then of course, eukaryotes, which include us. One thing I want to note is that we think of Luca as being the last universal common ancestor. So we think of it often as being the first, but actually it could be that Luca was the last. Luca could have been a survivor, and there may have been other branches on the tree of life that all just eventually went extinct. And Luca, the ancestor to all of us, was actually the one that somehow made it through that early, part of the evolution of life, and was the survivor, sort of like a Noah, if you will, on the early Earth. And it's kind of hard for us to really probe that far back. It sort of raises the question of how do we know what we know about the early Earth and early life? And how far back can our earliest memories go? What is the limit of our methods in trying to infer something about what the earliest life on Earth looked like? And that's something we always have to keep in mind when we're thinking about the earliest life on Earth. So just to give you guys a sense of how we actually learn something about what Luca may have looked like, I also, like many of you, am teaching right now, so I'm also really into the iPad doodling thing, so apologies for my artistic skills. But this is a rendition of, for example, if you have a collection of modern cells, these colored dots represent genes that might be in the collection of modern cells that we see today. And one thing we can do is look at what genes all these modern cells may have in common. Like, for example, all of these cells have a red dot and all of these cells have a purple dot. That suggests that these genes are universal to all of those cells. And if you use something called parsimony, that might suggest that you could infer that maybe the last universal common ancestor to all life on Earth may have had the red and the purple dot, or those types of genes. So that's a very, very broad 50,000-foot view at some of the techniques we use to look at modern life and try to infer something about what the earliest life might have looked like. And what I'm showing you here is a summary of one paper that did this, that came out a few years ago now, that used that method basically to try to infer something about what our last universal common ancestor may have looked like and how it behaved. According to this paper, which I should mention was controversial and is by no means the final word on this, but just to give you an example of what we're maybe learning, they argued that LUCA may have lived in hydrothermal vents, which would correspond a bit with what John was talking about where life may have first originated. It may have fixed its own carbon, meaning it may have been autotrophic. So it may have been like other organisms that are able to pull carbon dioxide or other inorganic carbon out of their environment and turn that into organic carbon to make building blocks and to get energy. They also argued that LUCA may have also fixed its own nitrogen. So it was really self-sufficient. It could maybe pull nitrogen gas out of its atmosphere, turn it into biologically useful nitrogen compounds, and then make building blocks from that. And they think, again, possibly, if you agree with this paper, that LUCA may have lived in a hydrogen and iron-rich habitat, which goes back to this idea that maybe it lived in something like hydrothermal vents. And I should say these vents here that you're seeing are at Picard, which is the deepest known hydrothermal system that we've heard of to date, I think. These are some really beautiful chimneys that we saw on a cruise this past January that I was lucky enough to go on, thanks to Julie, who's sitting in the audience right now. So that is one possible explanation based on, learning from genomic memories about what LUCA may have looked like. And then there's the question of how LUCA may have evolved over time. And one of the important things to know is that, to sort of continue with the themes that John was talking about, information and communication and information sharing were key for the evolution of early life. A lot of researchers agree that the way early life evolved involved a whole lot of gene sharing between different communities on the early Earth. We now call that form of gene sharing horizontal gene transfer. And it's a way to sort of share ideas between early cells. You could almost imagine early communities on Earth kind of tinkering with new quote unquote ideas as they swap functions and swap genes. And the ones that worked best were the ones that then continue to evolve and become the lineages that we know of today. What I think is really interesting about this is that just like humans, communication requires a common language. And that would have been the same for early life. And so it's been argued that, John talked about the unity of biochemistry, how all life on Earth shares certain types of biochemical processes and also a common language, which is DNA. And we needed to have that common language in order to start sharing these ideas in order for evolution to really get going. And I find that to be a really interesting idea. Just to give you a little more details on horizontal gene transfer and what that actually means. It's a method of acquiring genes from other members of a community. So instead of gaining genes from your parents, like we normally hear about in biology textbooks, this is a way for genes to be shared often between microbes or between viruses infecting microbes. So it can happen through basically bacteria or archaea or something taking up DNA that's just freely living in the environment, and they can pull that in and take that into their genome. They can share DNA through viruses. So a virus could infect one organism, and as it's making new viruses and repackaging DNA into the viral capsid, it can take up part of the host, its victim's DNA, put that into the virus capsid, infect a new host, and share DNA that way. So we can thank viruses for a lot of this gene transfer and gene sharing. And also bacterial sex is another way that genes can be shared between different types of bacteria. So when I say horizontal gene transfer, this is basically what I'm talking about, these three methods. And so what I've been doing lately is using this idea of horizontal gene transfer and gene sharing to ask more questions about the evolution of life on Earth. So just to give you a bit of a primer here, if a certain microbe acquires a new gene through horizontal gene transfer, there's basically two options, two paths that it can take from there. One is that if that new gene, upon being acquired, is actually useful to the cell, in other words, it helps it adapt and survive in its environment, that gene is often retained within that genome, and we can see this in genomic data. If the gene is not useful, oftentimes what we see in these populations is the gene is then lost in the population. That strain that had acquired that gene becomes outcompeted in the population. And so if a gene is acquired into a genome and it's not useful, it often isn't retained within the population. And the reason this is interesting is that we can learn something from this. We can figure out which genes have been useful for a specific population. And by looking into ocean memories, we can try to figure out when those specific genes became useful and use that to learn something about how cells have evolved over time. So like I said, to do this, we can identify when microbes gained new genes and kept them in our distant past, which might tell us something about their evolution. And to do that, it requires cooperation between those of us who are looking at memories and microbial genomes, so trying to infer things about ancient microbial evolution based on their genomes, and people like Patrick and others who are looking at memories in the rock record. We can bring those two things together and create a tree of life, kind of like the one you're seeing here, and try to pinpoint dates to it to say, okay, based on the rock record, we think oxygen may have arisen around the time this clade emerged. And we think that microfossils for the first eukaryotes arose around the time this clade emerged. And we can start to sort of anchor our tree to different points in time. And so just to give you an example of the kinds of stuff I've done, this is a paper that just got accepted last week, actually, where we were using these techniques to try to infer something about how microbes have been using nitrogen throughout the 4 billion years of Earth's history. And we can look at when did different types of genes in the nitrogen cycle, which is what I'm showing you here, when did different types of genes in that nitrogen cycle become more useful to these microbes over time? And can we see trends over time with when certain parts of the nitrogen cycle seem to be being shared among microbes more in Earth history? And what we show is that there's actually patterns here where we're seeing that certain stages of the nitrogen cycle seem to have been very useful very early on, like that step of taking nitrogen directly from the air and turning it into biologically useful nitrogen was something that we saw very early on in Earth history, which makes a certain amount of sense. You need to get that nitrogen from something. And then as Earth changed over time, as oxygen appeared on the planet, we start to see other parts of the nitrogen cycle become more, I hesitate to use the word active, but we were seeing more activity in terms of horizontal gene transfer later on in the history of life. The reason I'm interested in this is that, so microbial memories are revealing how life and the planet have changed together over time. And the reason I'm interested in this is because, what we're seeing is that within these microbial communities, we have these networks of shared genetic memory where genes are being shared and microbes are gaining new functions and sharing them with each other. And as microbes are performing these functions, they're releasing things into their environment. So their functions are affecting the environment around them. They're releasing gases, they're releasing metabolites that affect, say, the ocean that they're living in. These microbes are taking up metabolites, they're releasing waste products into the environment. So they're absolutely affecting the environment around them. We know that the reason we're breathing oxygen is because it's a waste product for some microbes and plants. Those microbial metabolisms then have an effect on the entire ocean and the entire planet because these microbes are everywhere, they're extremely abundant. And those functions that they have can affect the water column and the atmosphere and the entire planet's biogeochemical cycles. I'll note that part of the reason this project I've been working on, people are interested in it is that not only is it affecting the whole planet, but it's affecting our atmosphere, and you can actually see some of those signatures from space. And so I'm working with exoplanet scientists who are interested in knowing, can we see signatures of these microbes if we were on a distant exoplanet looking at Earth through a telescope, which I think is kind of amazing. Those planetary and ocean conditions, in turn, will affect microbial activity and affect the way those microbes adapt and evolve over time. So we're seeing this really intimate coevolution between what's happening on Earth in terms of the environment, the ocean, the rocks, the atmosphere, and then what's happening in terms of the microbes that are living on that planet and how they're affecting and driving each other and evolving together over time. And by looking at, like I'm saying, these genetic memories and these rock record memories, we can try to sort of piece together what's been happening on Earth history over time. So I'm going to end with another beautiful picture from the Salish Sea from last October, and I think we can open it up to questions. Oops.
Really, really great. Two really great and provocative talks. So I like that you, Rika, I like that you titled your talk Sharing Memories, which could mean anything, but it was very appropriate here. And then, and John's talk was a tour de force of what we should pay attention to, or what we should think about early Earth and its how it can help us understand ocean memory. So we had a few different comments in the chat a little bit earlier, and so I will pause my own several questions, and I'll go through some of those chat questions. So there were a couple questions about zircon and serpentization. I've never said that out loud before. Is there a way to sort of summarize that really quickly, John? I know these are pretty complex.
Yes, I can. Zircon is a
subjects.
mineral. The one that we're talking about was found at the Jack Hills, Australia in 2014. It can be dated to 4.4 billion years ago based on uranium thorium isotope data. That's how they're dated. Secondly, because of the oxygen type data, you can say that water was already formed. So that at 4.4, based just on this zircon data, you had crust and you had water. And it's very, very powerful. And that particular Jack Hill zircon really made the news.
Okay. And then you discussed serpentization a few times. Sorry, and from what I gather, I've heard that word 100 times. I've never said it out loud. But from what I gather, the relevance for ocean memory is that it's providing different elements or it's recycling different elements into the ocean, or I mean, there was a variety of things that serpentization does. Is there any way to sort of provide a concise, not really definition, but something that's very focused on ocean memory, like linking that thing, serpentization, to ocean memory?
First of all, the element part is from magma hosted. That's the one that produces over 40 elements.
That
The
was this, okay.
serpentinizing system is really a chemical reaction between a mineral called olivine, iron magnesium silicate, that when it reacts with water, it hydrates and produces basically a high pH hydrogen. It's that hydrogen that is really crucial in that it reduces carbon dioxide. It's involved in that. And we're just starting to, I think, get a sense of the wide range of organic compounds that are being synthesized by that. As I said, I've been very excited about the amino acid that was synthesized by that tryptophan. And when that paper came out on Nature, Nature asked me to write a Nature News on it, which I did do, and try to expand on the relevance of that to the potential for the synthesis of nucleotide bases, for example, et cetera. So I think these are ongoing research projects that are really big time. I also want to make one other comment. The origin of life community is extraordinarily divided. They're almost as bad as the Republican and Democratic Party right now. But,
really?
And there's many different ways in which the origin of life community is focused. The one group, which is what we call the metabolism first group, is really putting a lot of emphasis on these serpentinizing systems. And the whole concept of an alkaline system, high pH system, gives you a pH gradient, which they're using as a form of a way in which you can actually form energy, which is something that we actually see in living organisms. So there's a lot of things that are going on in here. And I'm not going to talk about the alternative views and the controversies, but going to an Origin of Life meeting is one of the most exciting things going on, because I have heard scientists get up and tell other scientists you're full of shit, and after their talk. And so it's just, I'm just, oh, yeah. So it's that kind of a community right now.
Yeah, that sounds like fun, actually.
It is fun.
Rika, we have a question from Ben. Is it accurate to say that what you described, the microbial genetic memory sharing, is that accurate to say that is like epigenetics?
Yeah, I saw that question in the chat, and that's a really fascinating question. As somebody who knows about as much about epigenetics as maybe the rest of you. So epigenetics is basically this process where you can methylate DNA in response to conditions that are going on during an organism's lifetime. So basically these are changes that can be inherited, but are not inherited from your parents. They occur during an organism's lifetime. At least that's my understanding of epigenetics. And what's interesting
about methylate, methylate, you meant turn on or turn off, basically, right?
Add a little CH3 to mark the DNA. And I think it can affect things like expression. And I guess what's interesting about that is it's a change that occurs during an organism's lifetime that can then be inherited to their offspring. And in that sense, yes, horizontal gene transfer is similar in that respect. And it's almost Lamarckian, that changes that occur during a lifetime can then be passed on to offspring, which is, not what Darwin had originally suggested. And then the question was from Ben, so I would say they're analogous. And how might we be sharing such deep memories similarly today? I don't know enough about epigenetics to know how long those signals last, and whether you could use that as a way to probe into deep history for humans as well. That's something that I don't know much about, but it's a really interesting question.
Jacob, did you want to ask your question directly?
Sure.
Instead of I can mangle it for you if you'd like, but
you can, we can both read it directly off the chat window. No, Rika, thanks so much for that talk. I was wondering you hinted at this. I was wondering if you elaborate a little bit more about how exactly, from looking at these different genomes. I know we're looking at relationships between genes and so forth, but how exactly do you estimate when in Earth's history a new gene emerged? Then how precise are those measurements? Are we really able to say, this happened at 3.6 billion and this happened at 3.7 billion?
Yep. So Jacob wants to get wonky. So excuse me for a second
yes,
while
please.
I show you a slide. Basically what we're doing is we're looking at species trees. So based on highly conserved ribosomal genes, like the hug tree that I showed you, and we're comparing those to gene trees of genes that we're specifically interested in, like the NIF gene for nitrogen fixation. By comparing the shapes of those two trees, we can identify gene duplication, gene loss, and horizontal gene transfer events. So that's how we're actually figuring out when these events occurred, or that's how we're identifying the events in the first place. And then in order to figure out when they occurred, that's where, yes, it's not that precise. So what you're doing is you're making a chronogram. You're creating basically a molecular clock. You're anchoring that with certain time points based on the rock record. And one of my close collaborators, who John mentioned, Eva Stuken, is an expert on early Earth processes and isotopic geochemistry. And she can give me like, okay, we think that based on the rock record, methanogenesis seems to have arisen within these bounds. And we can put upper and lower bounds on that within our chronogram. But you should see the size of my error bars. They are massive, right? And so, because when you're trying to go into deep time, like it's, we look at this with a huge grain of salt. And so when I'm talking to my students, I always say, let's emphasize not so much exactly when these things happen, because I don't necessarily believe it when you're looking that far back in time, because, the estimates from the phylogenetics gets a little dicey. The rock record itself gets dicey when you go back that far back in time. So we emphasize the relative timing around which different things occurred to try to get a sense of this seems to have happened first and then this and then this. And that's what we try to emphasize. But yeah, it's a really good question.
Awesome. Thank you.
when you talk about error bars, Rika, are you talking plus or minus 100 million years or
Oh yeah, definitely.
500?
When you get back that far, it's hundreds of millions of years. Yeah.
Okay, so that's quite a bit. I had some questions, but I'm gonna let people. I saw Jody asked if Rika uses the word memory in any of your papers.
So I saw that and I was trying to think back. I have definitely used it in abstracts that I have written ever since I got involved with this group. So it's definitely showed up in abstracts and talks that I have given. And I can't remember off the top of my head if I've used it in any of my papers. I don't know if Julie has seen me use it in any of her papers we've written together. I'm not sure that I have, but definitely abstracts and talks that I've given for sure.
Once it enters the literature, we're home.
How would you use it, Jody? Like, you know, I think, you know, we talk about adaptation and, you know, things like that. I know we haven't used it, Rika. Maybe we'll sneak it into the next one.
Well, I think the way Rika used it today was beautiful. Shared genetic memories for horizontal gene transfer. Perfect.
Yeah, and there are other examples of the word memory being used in oceanography. There aren't a lot, but my friend Francois Primo is one of the first that I could find.
Yes, in physical oceanography, there are quite a few. And there are some papers in ecosystem and whale studies that use memory. But in the microbial world, there are some microbial memory papers, but not from the ocean yet.
Well,
So you guys be first.
What I appreciate about using it within this group is from all the discussions we've had, I think there's an understanding that when I say memory, we're not talking about a record, right? Things are forgotten and things are reinforced. And I'm using memory very much with that in mind. And I know that this group has had that conversation and understands that, but I think that would need to be explained of putting it into a paper, that when we say memory, we mean like things that have been forgotten and reinforced over time, right? There's that important element to it.
Rika, could I ask you a question? This is going back to a conversation we had a year ago on the Rachel Carson in the Salish Sea, but I'm really interested in this idea of microbes, you know, having sort of a macro effect, and what you were talking about, how because microbes take up metabolites and release waste products, that their metabolism affects the entire ocean and planet. And so when you're working with planetary scientists, you're saying if you were to look down at Earth, you could see potentially, you know, life, microbial life in that way. And I'm curious if you could expand upon any examples about looking at other planets, and if this has been sort of posited, you know, looking at.
Yeah, that sounds like something John is probably excited to talk about too.
Yeah.
In order, or maybe together.
John, do you want to talk about biosignatures?
Either one.
Biosignatures. That's the term used in astrobiology.
Okay.
John, do you want to jump in and talk about it, or should I?
You do that.
He's handing it on to me. Okay. Biosignatures are basically signatures that we can see, usually using spectroscopy, looking at other planets, usually gases that are present in atmospheres. And there's been a lot of work done by people like Victoria Meadows at the Virtual Planetary Laboratory at UW, who's been thinking about, okay, what potential gases do we know come from life and not from other processes? And that's where it gets tricky, right? Is you have to distinguish what is known to come from life and what comes from abiotic processes. Like carbon dioxide, yeah, life gives off plenty of carbon dioxide, but so do volcanoes, right? So you have to be able to distinguish them. Oxygen is an important one. We do know that oxygen is produced from some abiotic processes, but mostly it's life that produces it here on Earth. In terms of the nitrogen cycle paper, the one she was interested in was laughing gas. Nitrous oxide is one that you can see from space. You can detect it with instruments, and we know that for the most part, it seems to be produced by life. The most famous one that was in the news a couple weeks ago is phosphine. You guys may have heard that there was this discovery that there was phosphine in the atmosphere of Venus. There's been a whole conversation about that, and whether, first of all, was that really phosphine? And second of all, if it really was phosphine, do we actually believe that only life can produce it, or are there other processes that produce it? So there are a lot of gases that are out there that we're looking at as potential biosignatures. both, you know, is it something life produces, and can we see it with telescopes? That's the other important part of it. But it's really
Cool.
fun.
We had John, I don't know if you want to elaborate on that, or we had a different question for you from
I'll just ask,
Stephen
just make a
Fiore.
little bit of a comment.
Yeah.
Because Rika is correct. But what we're waiting for is the James Webb Telescope, which will be coming out in another couple of years, hopefully. It's way, way delayed. And that has the potential to measure a wide range of chemistry in the atmosphere. And in some of the recent papers, even hydrogen and along with oxygen and you know, other kinds of gases that are there that might tell us something about whether or not life is there. Don't get me started on the phosgene thing. I have received more emails asking me what I think about the phosgene and Venus, and I think I've ever received on anything that's come out regarding astrobiology. So I've had to do some research on that, and I have to say, just to end it, that I'm extraordinarily skeptical about the conclusions of that paper in Nature Astronomy.
That was the feeling I got from. He asks, there's a lot of phosphine in chicken poop, so maybe there's a lot of chickens on Venus. That was a question, I guess.
No, sorry. You can consider me a skeptic as well.
Sounds like it. From the artists on board, unless there's more science questions from the artists on board. Oh wait, no, sorry. There was the one question from Stephen about. I'm gonna have to read it again. It had, John, in your talk, you specifically mentioned a quote global Earth. What the question was? What do you mean when you say that life arising possibly involved a global Earth?
Okay, this is something that also originally came out of the Eva Stucken paper that I talked about in 2013 and got expanded again in a book chapter that Rika and Eva and I just got published a few months ago. The concept was, in order to get the how question, remember I mentioned when, what, etc. The how question is the big, huge unknown question. How did life get started? How do we make the chemicals? How do we polymerize them? How do we make them into things like proteins and nucleic acids, etc.? And the only way that we could figure that out as a collective group was to take advantage of everything that's going on the global Earth during that late Hadean period that I was talking about. And because none of the hydrothermal systems themselves are producing everything that we actually need to make life. So the how question that we're really examining is, what are the full range of environmental conditions that are absolutely necessary to actually go from chemistry to something that's living? And no single environment I can think of can do that.
So the term global Earth meant that you need all of these systems working together to create the fundamental building blocks.
At the Hadean, which was primarily, you know, in that period, you know, while there may have been some islands and that, they would have all been serpentinizing type systems, highly mafic. But we're really talking about what the crust down into the magma really looked like. There would be tons of gradients in that global crustal system with temperature, pH, salinity, wet, dry, everything going on. And so that's what's really needed to get all the things that we need to get. If I were to expand on this in another talk, I would talk about what minerals we are that are known to actually do some of these things and where you find them and when they came about, et cetera. But that's another talk.
Okay, I see Melody has a comment. I just wanted to put out a comment to the artists. Not a comment or a question, you know, do you have any specific inspiration from seeing these talks? I know that I learned a lot from these talks, and I've been in you know, involved in this for some time. At least I've heard the terms, even if I haven't spoken them out loud. Any artists want to comment, or if not, Melody, you could maybe ask your question out loud.
Okay, I'll just pitch in a little bit here. I know one of the things the group has talked about in the past a lot is the, which definition of memory we kind of go with, and how what ocean question we start with leads to maybe a different formulation of that. So for example, Rika's distinction between a sense of recorded memory versus memory involving things forgotten and reinforced is really interesting, because as someone who thinks more about literature and philosophy, this raises some interesting questions over memory for whom. So not just memory out there, but memory for that's useful for a particular community. So I thought that was interesting. But one of the questions I bring is, how does the ocean make memory studies ask different questions, rather than what does memory studies contribute to the study of the ocean? So it's something I've wanted to kind of flip with the group, because I think that the tendency, at least in the humanities, is not to think about the ocean, which is why I like to go there and love listening to all the science presentations. So one I think promising implication from the both talks today is to ask, what are these narratives about ocean memory contributing to memory studies, specifically around awareness of environment? So for example, in Rika's talk, what would be the implications of thinking of memory as not just something interior, but something that produces these effects we live in, like oxygen? I mean, I think that that's a really interesting question that mixes up the inside and outside in ways that aren't super stable. And I think it's interesting we get to that through this question of microbial metabolism. So if memory has been, sort of usually been thought of something as deeply interior or contained, the microbes, of course, are doing the horizontal gene transfer, and then also having all these exudations or waste products that then becomes significant for other things. So I really think there's something here that's related to memory's production, or sorry, I guess you could say something like memory's production of an atmosphere that then becomes significant for other reasons. So that's my small riff on that.
What I like about that is, when we think about microbes sort of like sharing memories and then creating new functions, and then, having an effect on the atmosphere, the oxygen example being the most obvious one, that nearly killed everything on Earth. It was a total accident. And then it was toxic to most organisms that existed. And so it nearly killed everything. But then all of a sudden it enabled this new metabolism that we could get even more energy from, enabling animals like you and me to live today. So it was a total accident, nearly catastrophic, but then ended up being hugely beneficial. And I just find that dichotomy sort of fascinating.
Well, that's a really interesting way to think about ocean oxygenation. Tim, you want to ask your questions?
Yeah. Rika had brought up looking back at Earth from with satellite data, exoplanet as a exoplanet biosignature kind of context for looking at ourselves. What I wondered is there a are those biosignatures of Earth utilized, and how are those utilized, and are they openly available? So you'd mentioned that of our own biosignatures. So it's kind of like looking back at us. So I'm assuming that might be some kind of standard that we're reverse engineering, right? So instead of just looking outward. So yeah, I don't know what you what you're thinking about that. I've been thinking a lot about biosignature sonification lately. So, but yeah.
Wait, so you're wondering if the data, the spectral data is available that could be sonified?
Yeah, it definitely can be sonified. Anything can be sonified.
Yeah.
I don't know if it makes sense. So, but you had mentioned satellite data or biosignature data of the Earth. So is that, I just started looking for it, and there is some registry of it, but I didn't know if that's like a standard that is gone back to a lot.
I don't know. But I have a whole Slack channel of exoplanet people that I talk to. So now I'm going to ask them where one finds that kind of data. Because I just normally make my data and I hand it to them. So I can ask them and then tell them where we can find it.
Yeah. But yeah, so emphasizing, as you went over segments of the spectral data, the sonic print that would accompany it could be reinforcing as well to add another sensoria to that. So, and then I was also really interested in you doing astrovirology. I don't know. I was reading that about on one of your bios a little while ago, and I was like, yes, I want to know about that. So I just need to dig deeper into your papers, I guess, about that. So
well, that came from my time with John as a PhD student. He and I were talking a lot about like early viruses and how they could have facilitated communication between the earliest cells, because viruses are on the one hand a parasite that could destroy you, but on the other hand are these vectors for information, and so they're useful in that respect as well. And so we all think of viruses as being this bad thing, especially right now, but they also have benefits, which I think you guys all talked about for that Scientific American piece too.
Yeah. So yeah, but I might contact you about that because it sounds very compelling. Like, of course.
I think one of the most compelling parts about this is that every life form that we know of has a virus. And so if you talk about the origin of life, you're talking about the origin of viruses. So life and viruses are one and the same, it seems to me. That could be debated, I'm sure. That's new in the astrobiology community, the idea that we have to be thinking about viruses all the time.
Yeah. Excellent.
Just adding to what Tim said, I was thinking the same thing about like being able to see these visualizations or this data of, the biosignature of Earth, and I think one of the things that's been fascinating for me being part of this group is sort of over the last 3 years, realizing that microbes basically mediate everything, all processes. And I love this way that they're amplified, and being able to see it from that view would be really interesting. So anyway, Patrick, do you think we should?
We're over the 1 hour mark.
Over time. I think that there's some really interesting things happening here that we could maybe switch to Slack.
Yeah, we shouldn't force everyone to be here, but this is exactly like ocean memory, right? Like we're just starting. It feels like we're just getting going. Hopefully we'll have a live workshop in the next year, right? We haven't talked about that, but was hoping this spring, but it doesn't look so hot. I guess
our next event will be virtual as well.
Boo.
Our next seed seminar will be virtual, but perhaps our third one will be in person.
Yeah. Well, this was fun. I'm glad we had a pretty good turnout. And I guess everyone should just await our next email about the next webinar. We don't have people in mind yet, but we'll.
Yes, actually, we do. I think it's Jody.
Did I miss them?
Yeah, I think it's Jody.
Oh, we already have that.
Yeah.
Oh.
I think it's Jody and I.
So we should advertise that then. Okay.
But you better double check with your invitees before you advertise.
Yeah. Yeah.
Okay.
I wanted to also thank Kathy and Jacob, who've been on our organizing team as well. So thanks to them, and thank you so much to John and Rika for really amazing talks.
Yeah, actually, this really inspired me. The horizontal gene transfer is already kind of putting inspiration into some of the art pieces that I'm doing. Cool. So
I would love to see that.
Yeah, just the idea of the patterns of sharing within the active microbial memories of life of all of the bodies that were put into the ocean over the 400 years of the slave trade. That, it's interesting to think about. So thank you.
All right, that was great, everyone. See you in a month or so after we check in with our next proposed speakers. Do we have a date, or am I?
We just had a date of November, but maybe not a specific,
Okay.
like
I think
mid,
we were talking about doing the third Thursday,
right, yeah.
second Thursday, so we'll have to look back at our notes, so.
Isn't that like Thanksgiving?
I was
Anyway,
just
we
gonna
can have
say,
this
yeah.
discussion. Thanks, everyone.
Thank you.
Thank you.
See you in a month.
Good to see
Bye.
everyone.
Okay.
Bye.
Bye, everybody.
Thanks, you guys. Bye.
Okay,
You all good, Max?
Yep, I got it.
got everything.
Okay.
Yep.
And then I guess the next one, we'll have, oh wait, Kathy, if you're speaking at the next one, you can't do these host duties, so that's fine.
Well, the host duties for me were just sending out the invitation, really. It wasn't,
Oh, I mean, for the next one.
yeah,
But that's fine.
I can still do that. I'm
Yeah.
not I mean, it's easy for me to do a calendar invite. So
Cool. Well,
I
it
just
was,
have
you
to
guys were,
meet with Jody, I guess, and figure out what we're going to talk about. So
yeah, that's
I will send
very
her an email. Probably tomorrow.
And I was thinking, when you mentioned horizontal gene transfer inspiring your art, the whole our whole proposal with Chris is like horizontal gene transfer. We're literally moving a thing to each other and like
Right.
taking what we like. That could have been, we could have wrote that into the proposal, but I guess we'll write it into our final report.
Yeah, it can evolve.
I'm pretty stoked. I mean, like I said, I'm all of a sudden feeling relieved of some backlogged work, and so maybe I'll put some pressure on Chris to start this thing going. I wanted her to start it off.
Okay.
Seems right. I don't know
Yeah,
why.
no,
I guess because her and I have been talking about it for years.
No, it makes total sense. And
Start the gene transfer.
Yeah, no, I have a class that starts for like 5 weeks that I'm taking in addition to teaching, but I'm still
you're teaching too.
Yeah, I'm TAing for a film class, but it's not as demanding as my last TA-ship. So
good.
Because I've been working on art, because Max and I are on the other grant project that I'm doing. So
oh really? Okay, that's why you guys were talking.
Yeah,
this is the diving thing.
Yeah, this is descent and transformation. So I'm working on that a lot too.
Yeah, it's not the diving thing. It's the cross media. What is it? The multimedia.
Multi-channel.
Multi-channel
video VR installation.
Yeah.
Really?
Yeah.
What that sounds. I don't know what that means, but it sounds a lot cooler than the diving thing.
No, it
You're
involves.
right.
Yeah.
I undersold it.
It involves the
I didn't
dive.
see anyone else's proposals, so I don't know what else is out there.
Oh, okay. So yeah, because I didn't know who was on what committee for. Do you know when they're going to send out the press release about our grants?
There's a press release? I don't know.
Yeah, remember?
I didn't know there was going to be.
Yeah, they asked us for emails about who we would want to receive our announcements.
That happened? I don't remember.
It was part of the congratulations.
Past couple months are a blur.
Yeah, it was
really?
part of the congratulations, you've received a grant email.
Oh, I didn't write anything.
So, oh well. Yeah, I finally had to tell people at school because it involves like funding and stuff. So
yeah, I should probably. I don't know how to do that with this. I've done it with other much larger grants.
Well, supposedly they're going to send the announcement to whomever you indicated you wanted to receive it. So.
Okay, I probably.
And it's a way of like promoting you and what you do. That's why. Are you going to go to that meeting with the Humanities Center on the 20th? I think it is.
It's in my calendar, and then someone else, totally independent of our communication, asked me about. She said, "Is this the thing you were talking about?" Because I sent it off to the oceanographers, the the Year of the Ocean, I think you called it.
Right.
So there's at least one other. I don't know why there isn't more excitement, enthusiasm about it. I mean, everyone's, I think, overwhelmed with life.
Right.
But yeah, I have it in my calendar. I was planning on attending
Okay.
for
Yeah.
sure.
I will definitely see you there. So do you think it's okay for me to not wait and go ahead and contact Jody and say, you know,
Oh, for sure.
okay, good.
Yeah.
I was gonna
Yeah.
pretty much do it after this. So, because I wanted to see
think
how this went.
Okay. Yeah, I think it would. Okay. I mean,
Yes.
the only worry, I know Rika and John both pretty well. And so
Mhm.
the only worry was that John was going to speak too much because that's his history.
Right.
The third, I was never worried I was going to be it was always, I always knew it was going to be interesting. By the way, the third Thursday of November is fine. Thanksgiving's the fourth Thursday.
Okay.
So that would work okay if you can arrange for that time with Jody and
Mhm.
talk about ways in which you can sort of interlock what you're going to present. That would
Yeah.
be great.
Yeah. I just have to figure out how I'm going to present what I think I want to present because I'm not a scientist. So.
No, I can't wait. I want there to be, yeah, I want to see this sort of art science thing going on.
Okay.
Maybe we should let
Okay.
Max go.
Yep.
I'm gonna head out.
Okay.
Okay.
Good
No,
to
I'm
see
gonna
you
go
all.
too.
Thanks very much.
Let me
See
know
ya.
if you need anything. I'll be in the emails and
Okay.
always available.
Okay.
Thanks, Max.
Great. Alrighty. Bye.
Bye.